The Botany of BC Mountains project is a long-term collaborative research venture initiated in 2002 by Royal BC Museum botanists Ken Meyer and Richard Hebda. I joined the project in 2012, and I'm thrilled to be part of this focused and sustained effort to document the alpine flora of the province. Our external collaborators are Jerry Allen, Professor of Biology at the University of Victoria, and Will Mackenzie, Provincial Research Ecologist with the Ministry of Forests, Lands and Natural Resource Operations. This morning I'll give you an introduction to the project as a whole, and then present preliminary analysis of our collections data over the first 10 years of the project. Ken will follow up to present case studies of 2 alpine plant species that highlight a newly described pattern of distribution that provide clues about the history of the landscape of BC. Worldwide, the alpine biome, the land above the trees, accounts for only 3% of terrestrial ecosystems. In British Columbia, however, more than 12% of the provincial land base is classified as alpine tundra. This makes BC one of the most mountainous jurisdictions in the world and a key steward of global biodiversity. As a result of climate change, tree lines are ascending and large areas of alpine habitat are at risk of being lost. Never has it been more important to document these fragile ecosystems and the flora and fauna that are adapted to life in these extreme high-altitude environments. In 2002, Royal BC Museum botanists began to focus on improving knowledge about this fragile biome by making comprehensive collections of vascular plant species. Pink dots on this map indicate the mountains we have visited between 2002 and 2017. And I'll tell you a little bit about the rest of the map. Based on differences in biology, geology, and climate, the alpine biome in BC is classified into 3 zones indicated by the blue, green, and brown shading on this map. The boreal Altai fescue zone is in blue, is the most widespread, covering most of northern BC and running south along the leeward side of the Coast Mountains. Coastal mountain heather zone in green runs along the windward side of the Coast Mountains, and the interior mountain heather alpine zone is in brown, runs along the Rocky Mountains. As you can see, RBCM collections and research to date is largely focused on the BAFA zone, the boreal Altai fescue in blue. This is because the mountain ranges in this zone are woefully understudied. We have 3 main objectives for this project. First is to document the alpine plant diversity in BC by making comprehensive collections. This means that we add an 11 by 16 inch herbarium sheet to the botany collection for each species we collect on each mountain. Based on more or less standardized approach, we find between 100 and 200 species per mountain. To date, we have added over 18,000 specimens to the Royal BCM botany collection, and most of these are now available for study by researchers around the world. By 2022, the collecting phase of the project will end, and this number will be as high as 25,000 specimens. Over the next 5 years, field sampling will be completed. In 2018 and 19, we'll complete sampling of the BAFA zone in the northern Rocky Mountains and on the leeward side of the Coast Mountains. We'll then make comparative collections at the intersection of the 3 zones in 2020, and then sample the IMA zone, the interior mountain heather zone in the southern Rocky Mountains of Canada for 2021 and 2022. Our second objective is to identify the environmental drivers of alpine plant diversity. We are asking the question, what environmental factors control which species grow on each mountain? Does the underlying bedrock or soil type play a role? Does the timing and level of seasonally available moisture control what plants grow where? Is it a mix of these? Another question is, why do some mountains harbor more species than others? Do environmental factors control the number of species too? Or are very diverse mountains simply at a crossroad of floristic influences, where species common to higher latitudes are found growing with species common to lower latitudes? The application of geospatial analysis and other statistical tools to a very large data set such as this one is at the forefront of biodiversity science and is a field that museum collections contribute to directly. I'll present a preliminary analysis on the next slide. Our third objective is to test alternative hypotheses about the history of the BC landscape. In particular, we are using variation in DNA sequence of several widely distributed alpine plant species to look for evidence of large glacial refugia, that is to say, places on the BC landscape where plants and animals may have survived the last ice age. This part of the project is largely complete, and results have been published in 6 peer-reviewed articles to date. Ken will present evidence from one of these species in his talk. So now let me just show you some preliminary results from an analysis conducted by Will Mackenzie, who's the spatial statistician on our team. We are in the data exploration phase of this work, meaning we're assessing the structure, the size, the quality, and the limitations of our data. As field sampling is completed over the next few years, we'll continue to add information and arrive at some point at a final data set. This figure is a graphic representation of the similarity among 53 mountains visited by us between 2002 and 2012. And by similarity, I mean how closely the list of species from one mountain matches the list of species from another. This figure resembles a phylogeographic tree showing evolutionary relationships among organisms, and we can read it in essentially the same way. Individual mountains are at the very end of the branches, obscured by the color boxes. Think of them as the leaves on a tree. As we work away from the individual mountains, the structure of the branching shows relationships among small groups of mountains and eventually among large groups of mountains. I've simplified the diagram to highlight the main clusters so we can talk about broad patterns. We see that there are 2 main branches in this diagram, this branch here and this branch here. So these clusters I'm calling cluster 1 and cluster 2. Cluster 1 is then split into 2 branches, which I'm calling cluster 1A and 1B. As our data set grows, more leaves will be added and the branching pattern may change significantly. So now we have a sense of how individual mountains fall into groups and how these groups are similar to each other in an ascending hierarchy. The next step is to visualize this information spatially by plotting the location of each of these groups on a map to see if there is a discernible pattern. At first glance, we can see that there is some geographic structure to the distribution of each cluster. That is to say, there are areas on the map where only one color occurs. The exception to this is the placement of yellow mountains clearly in the territory of otherwise blue or green mountains, up here in the north and down here in the south. These outliers from the general pattern could indicate errors in the data, errors in mapping, incomplete data, or may in fact represent reality. Visualizing information is a critical step in the data exploration phase of this research and suggests we need to return to the data at these mountains. Ignoring the yellow icons for a moment, we can see that there is a line delineating the range of green and blue mountains. What does this line represent? Is it a mountain range that acts as a barrier to dispersal to some species? Is it underlying geology on either side of this line significantly different? Is there a moisture gradient that hits a significant limit at this line? Our goal going forward is to identify all biogeographic breaks, this is a biogeographic break, such as this one within the Alpine dataset. For each biogeographic break, we can then use the tools of geographic information systems to look at correlated breaks in environmental factors such as geology, rainfall, temperature, etc. Okay, so next steps for this geospatial analysis include adding collections from 2013 onward. We need to develop an index of effort that will add an estimate of the number of person days spent on each mountain. That will help us control for the species number, the variation of species number that is based on effort. Importantly, we need to test for environmental correlates that might explain some of these geographic distributions. It is no mean feat to maintain and fund a sustained effort, such as this one, and the Royal BC Museum is proud to make this lasting contribution to biodiversity science in Canada. We have built a globally significant collection that will serve as a unique biodiversity baseline as we track the effects of climate change on the natural and the human world. And finally, a special thanks to Will Mackenzie, pictured here, the lead on this part of the Botany of BC Mountains project, who also conducted the analysis I presented. Thank you.